Hi everyone, thanks for checking out this video. So I'm actually making this video in response to a request that I got from a viewer. Someone asked to hear about the toggle switch model of gene regulation and how to write a stochastic simulation of it. So I'm going to try to cover this topic in this video. So in the toggle switch model, you have two genes that both negatively regulate each other, or in other words, they repress each other. So I modeled that here in the figure with these like lines with these like kind of bars at the end. In when you're making a diagram of gene regulation, this usually means negative regulation or repression. So in this case, we have gene 1 and gene 2, and they both negatively regulate the expression of each other. So I'll just tell you guys like the basics of the model, and then we'll get into the code for it. So we have gene 1. It's produced with the rate K1, but the whole production term is also going to incorporate negative regulation by gene 2. So we'll work that into the math in the next part. But it has some production parameter here K1, and it produces a protein, and the protein then decays with rate gamma 1. And that's what this means here, by the way. This circle with a line through it just means like the degradation of whatever we're talking about. So yeah, it's produced and then it degrades with some rate gamma 1. You guys may notice that for the purposes of this video, I'm actually skipping over the RNA step. I'm just saying that the protein is made with some rate from the gene. If you guys want it to be more realistic, you could include the RNA step here and then the RNA to the protein. But just for the purposes of making like a simple tutorial, I'm actually skipping over the RNA step. And then this protein produced from gene 1 negatively regulates gene 2. And gene 2 also is produced with some rate K2, which is going to also be negatively regulated by gene 1. And then it decays with some rate gamma 2. So yeah, it's kind of symmetrical. Both of them are produced and both of them decay, and then both of them negatively regulate the other one. And the kind of dynamics you get with this system it's called a toggle switch, because basically if one of these genes is turned on or like currently being produced, then it'll negatively regulate the other one, so the other one will be turned off. So it's like flipping a switch when you have like a stochastic simulation of the system. It's like if one of them is on, the other one is off, and vice versa. So that's kind of the idea of like why it's called a toggle switch. Yeah, so that's a basic explanation. So next let's take a look at the math, and then we'll get into some of the code of it. Okay, guys, so it's like I said before, we're going to be making a stochastic model of this system, not an ODE model. But before we get into the stochastic model, I do want to show you guys like what the ODE system would look like, just so we I mean, sometimes I think even if you're not doing an ODE simulation, it helps to look at the ODEs first, or at least what they would be, so that you can make sure you're setting up your stochastic simulation correctly. So we have two ODEs here. There's one for gene 1 and one for gene 2. And by the way, guys, I realize I'm using the terminology a little bit sloppily, but when I'm talking about gene 1 and gene 2, what I'm really talking about is the protein products of these genes. So when I'm talking about like the level of gene 1, I'm talking about the level of the protein being produced by gene 1, and same for gene 2. I'm not talking about literally the nucleotides in the DNA themselves, talking about the protein here really. But yeah, so gene 1 is produced, it has some like production constant parameter K sub 1 over here. But this is also negatively regulated with this Hill function here. And if this looks unfamiliar to you, I'll put a link in the description to my video on Hill function, like negative Hill function repression models. But basically, if you have one gene negatively regulating another gene, you can model it with this this thing called a Hill function, or a Hill coefficient. And so we have gene 2 in the denominator here, and then we have some constant parameters C and H. And basically, this whole thing, as you get more and more of gene 2, this whole term becomes smaller. So basically, the more you have of gene 2, the smaller of a fraction this term becomes, and it means you're getting less of gene 1 produced because gene 2 is negatively regulating gene 1. So the more we have of gene 2, the lower the production of gene 1 is going to be. So that's kind of this whole production term here, just this whole hill term multiplying the k sub 1 production term. And then, like I said, we have gamma 1, which is a degradation term multiplied by the level of gene 1 for our degradation. And then basically it's a symmetric system. So basically our whole equation for the ODE for gene 2 is just the same exact thing, except that it's gene 1 repressing gene 2, and then K2 instead of K1, and then gamma 2 instead of gamma 1. I kept the same hill parameters here, so the same hill parameters C and H are in both gene 1 and gene 2. You guys could make these different here if you wanted to, but I just think it's easier for now to just keep them the same for basically just trying to make this tutorial as simple as possible. Okay, so that's what the ODE system is, but like I said, we're not simulating the ODEs, we're making a stochastic simulation. So when we do a stochastic simulation, hopefully you guys kind of already know this if you've watched my other videos. If you haven't, I'll put a link in the description to my video on the Gillespie algorithm, but assuming that you guys already kind of know the basics, I'll just give a quick review here. So in order to use the Gillespie algorithm to make a stochastic simulation, we need to make one of these event propensity tables. So we need to think about in our system, what are all the events that could be happening, and then what are all the propensities of those events. So there's basically 4 events that can happen. We can have a production of gene 1, we can have a degradation of gene 1, and then we can have a production of gene 2 and a degradation of gene 2. Again, I'm talking about the proteins that are being produced by these genes, not the literal segment of DNA themselves. And so for the event side of the table, for each of these 4 events, we need to write down what exactly is happening in the system when the event occurs. And for the production event for gene 1, what's actually happening with this event is the level or the count of gene 1 is being increased by 1 when there's a production event. And when there's a degradation event for gene 1, the count of gene 1 is being decreased by 1 because one of the actual protein molecules is disappearing. And then same thing for gene 2. When there's a production event, the count of gene 2 increases by 1. And then when there's a degradation event, the count of gene 2 decreases by 1. And then for our propensities here, we basically just copy and paste in these terms from the ODEs. So the propensity for the production of gene 1 is just this whole thing here, the whole production term. We just copy and paste this whole term into the propensity side of the table for gene 1. And then same thing for the degradation term, just copy and paste this whole thing into the propensity side here. And then we just do the same exact thing for the gene 2 events. Okay, so basically in order to do the Gillespie simulation, we need to make this event propensity table. And then from here, the actual code is actually not that difficult. Sometimes the hardest thing with these models is just defining the model itself and writing down this event propensity table in a way that actually makes sense for the system you're working with. But once you've been able to correctly write down a table like this, it's actually not that hard to write the Python code for the actual simulation. So let's get into that part next. So we'll actually do the Python code now. Okay, so we're going to be importing 3 libraries. We're going to import numpy smp, import matplotlib.pyplot splt, and then import random. Okay, so I'm hoping that you guys have watched my previous videos on the Gillespie algorithm in Python. And again, I'll link to the description to those videos if you haven't. But I'll kind of quickly review here. So when we're doing these simulations, we're going to keep track of the counts for our variables in lists. And basically, we're going to have a list for both of the variables we're keeping track of. They're both going to start off at 0, but they're going to be updated with new counts for these variables as the simulation progresses. And then we're going to have a count for the time too. Sorry. And basically what's going to happen in the simulation is we're going to go through a loop, and as different events are occurring, we're going to be appending to these lists. So we're going to append like different counts of these variables we're keeping track of. Like they're going to be updated, and we're going to keep track of their levels by appending to these lists. And then while this is happening, we're going to also be appending like time points too. So it's going to end up looking something like this. And we're going to, basically every time the loop iterates through, we're going to append one new thing to each one of these lists. And then even if it's staying the same, like maybe they'll all just be, I guess that doesn't, yeah, like sometimes there's going to be like staying the same too. You know what I mean? So, but we want them all to be, the list to all be the same size at each one of the loop iterations. So basically, just to keep track of the variables, we're either going to append a new value or we're going to append like the current value again. And then we're going to also keep track of the time by appending to this list. So that's kind of like the structure of how we're actually doing the simulation. Before we actually get started with the loop, we need to like define a couple things too. We need to define the endpoint of the simulation. So I'll call that t-end. Set that to 1,000. Just arbitrary time units. Could be, I don't know, seconds, minutes. I don't exactly know. It kind of depends what kind of system you're working with. But for now, I'll just make it like arbitrary time units. And then we need some values for our parameters. So k1, make that equal to 1. Gamma 1, 0.2. You guys can make these parameters like whatever you want. Maybe play around with them. See what happens when you change them up. And for now, I'm actually going to make k2 and gamma 2 just the same as k1 and gamma 1. Also just make them 1 and 0.2. So those are our constant like our constant parameters. We also need these hill parameters here, h and c for the hill function. So I'm just going to make c equal 1 and then h equal 9. Again, I encourage you guys to like play around with different parameters, change these up, see what happens when you mess around with them. But yeah, I'm just making these kind of this for now. Okay, so our actual simulation is going to occur within a while loop, and we're basically going to say while the most recent time point is less than t-end. Basically, we're going to keep doing the simulation as long as the most recent time point we've run into is less than our end point. That's going to tell the code to keep going with the simulation. And what we need to do here, like first of all, before we even do anything else, we need to put all of our propensities into a list. I'm going to call this the props list. Actually, I'm going to copy and paste. Yeah, actually, sorry, I'm just going to be a little bit lazy here and just copy and paste some of this. But then I'll explain what it is. So basically, just putting this whole thing in a props list, because basically, like the propensities, they're changing as the simulation progresses. So these propensities depend on the current levels of gene 2 and gene 1. So depending on what the current level of these variables, what the levels are, the propensities are going to be different. And the propensities are going to be like changing as the simulation progresses. So we need to recalculate what these propensities are at each iteration of this loop, basically. So each time we go through the loop, we recalculate this propensity list, and we're basically having like one element in the list for each of the propensities. So we have our first one here. This is the first propensity. I basically just typed in this propensity from the table here, but I'm making sure to use the most recent value of gene 2. Because, yeah, again, it's changing as we go through the simulation. We want every time the most recent value of gene 2 in the denominator here. But yeah, other than that, I copied and pasted this propensity right from the propensity table right into the propensity list in our code here. And then same thing for the degradation propensity. Copy and pasted right from here, this term here, copied and pasted, I mean, not copied and pasted, but like typed it right into our propensity list here. Again, the most recent value of gene 1, we're getting that with this index -1 of the gene 1 list here. And then basically the same thing for the gene 2 production propensity. Typed it right from here right into the list, and then again for the gene 2 degradation. So yeah, we just need to get all these organized into a list, and then we also, like, once we have them all in a list, for each iteration, we want to get the sum of the propensities. So we get this just with sum of the propensity list. And then, okay, so basically the Gillespie algorithm has 2 parts. So like the Gillespie algorithm, basically at each step of the simulation, you need to randomly choose 2 things. So you need to first randomly choose the time when the next event is going to occur. And then after you know the time of the next event, you need to actually randomly choose which event that's going to be. So the first thing we're choosing is the actual time point of the next event. So to do this, we're going to take a draw from a exponential random distribution. So we get that with numpy.random.exponential. We're going to set the scale to be 1 over the prop sum. For now, I'm not going to go deep in like the math of why we're doing this, why are we using an exponential distribution, and why we're setting this scale to be 1 over prop sum. Yeah, like maybe in the future I'll do a video going like more in like the math of and like the probability theory of like why we're using an exponential distribution, why we're setting the parameter this way. But for now, I'm just trying to make like a video to show you guys how to actually implement the simulation. So I'm going to kind of leave the math details off to like possibly a future video. But for now, you just need to know that like this tau, drawing from a random exponential distribution, this is going to be the increment from the last time point of the last event, how long it's going to take for the next event to occur, and we're choosing that randomly at each iteration of the loop. So after we have that, we're going to say t.append the last time point plus tau. So we're basically saying the time point of the next event is going to be the time point of the last event plus this number tau that we just randomly picked from this exponential distribution. Okay, so now we've chosen the time point that the next event will occur. So now we need to choose which event that will actually be. Remember there's 4 events. So we need to pick which event that will actually be that's occurring at this time point we just picked. So to do this, first we're going to take a random draw from a uniform distribution between 0 and 1. So I'm going to say random.uniform from 0 to 1, just a random decimal between 0 and 1 uniform distribution. And then we're going to use this random number to randomly pick which event we're going to be choosing. And so I'm actually going to be a bit lazy again and just copy and paste some code in here, but I'll explain it to you guys like what the code's actually doing. Okay, so we have a big block of code in here. So basically, based on this random number we just picked that we're calling rand, we're basically going to we're using this random number to take a random choice of which event we're choosing. Basically just a random draw between all 4 of these events, but each event, the probability of it happening is going to be weighted according to the current level of its propensity. So a way to think about this is that the probability that the next event is going to be this gene 1 production is going to be the current propensity of gene 1, the current value of this whole propensity, divided by the sum of the current values of all the propensities. So it's basically like just like taking a random draw of which event we're picking, but weighting all the probabilities accordingly. So yeah, gene 1, the probability of this being the next event is its current propensity divided by the sum of all of the propensities. Same thing with like the probability of like choosing the gene 1 degradation event is this current propensity of the gene 1 degradation divided by the sum of all of the current propensity values. But the way we're actually writing this into the code, I know it sounds simple. Hopefully sounds kind of simple the way I just explained it. It's just like choosing the probabilities of each of these four based on their current propensities divided by the total propensities of all of them. In order to actually write this into the code, basically that's what these if statements are doing. The if statement is just checking like where this random number, basically we're multiplying this random number we picked by the sum of all the propensities and just seeing like where that falls according to the consecutive summation of all of these propensities. So we basically multiply this random number by the propensity sum, and we say, okay, is that less than? Sorry, I actually just noticed a mistake here. These should be less than or equal to, not just less than. But yeah, so we say, is that less than or equal to this first propensity here? And if the answer is yes, then we choose this first event. And then if the answer is no, then we say, okay, well, is it less than or equal to the sum of these first two propensities? And if the answer to that one is yes, then we choose the second event. And then if the answer is no, we say, okay, is it less than or equal to the sum of these three propensities? And if the answer is yes, then we choose this third event. And if the answer is no, well, then we say, then we already know it's going to be the last one, but I just put it in the code. I wrote it as if we have like possibly more than 4 events and we wanted to keep adding events. Then we would say like, okay, is it less than the sum of these 4 propensities? And then since we only have 4, like it must be yes. But yeah, so that's basically. I hope that made sense. You may need to like wrap your brain around it a bit. I try to explain as clearly as I can, but yeah, we're just checking where the random number times the propensity sum falls according to like the consecutive summing up of these propensities. And if it's less than or equal to the first propensity, we choose the first one. And then basically what's happening in this block of code here is we're updating the variables accordingly based on choosing like which event we choose. So if we choose the gene 1 production event, we update so that the new value of gene 1 is the latest value plus 1 because we're increasing it by 1. And then we append to the gene 2 list just by appending the latest value of gene 2 without changing it. Same thing if it's a gene 1 decrease. We update, we append to the gene 1 list the latest value minus 1 because we're decreasing the level of gene 1 by 1. And then we append the latest value of gene 2 to the gene 2 list without changing it. And then basically same thing happening for the gene 2 production and gene 2 degradation events here. Again, I hope this makes sense. I know I'm kind of rushing. If anything doesn't make sense, I'll also link you guys to my like previous videos on the Gillespie algorithm and Python implementation. But for now, kind of like hoping that you guys have seen this before and already kind of know. Also, any questions, of course, just let me know in the comments and I'll try to help. But yeah, so basically this is the whole simulation. This while loop is the whole simulation. It's going to keep running through this, keep choosing events and keep updating until our latest time point becomes more than greater than or equal to our t end, and then it's going to stop. Okay, so yeah, I mean basically this is the whole simulation. So the next thing to do is just plot the results. So again, I'm going to be a little bit lazy here and copy and paste in some code. Just using matplotlib to make 2 subplots here, plotting these results for gene 1 and gene 2. Yeah, actually, I don't even need this part here. Okay, so yeah, let's just give this a try and see what it gives us. Yeah, so basically looked like how it should work. We can see that it starts off with gene 1 kind of turned on, and then gene 1 like just stochastically switches off and goes off, and then while gene 1 is off, gene 2 can then turn on, and then gene 2 kind of sustains itself, repressing gene 1, and then eventually gene 2 just turns off, and then gene 1 turns back on. But like the point of the toggle switch is that when one of them is on, the other one is off, and vice versa. Basically, you can only really have one on at a time because they both repress each other. So that's kind of why it's called a toggle switch. But again, this is all stochastic. So if we run it again, we're going to get a different result. Yeah, so running it again, this time it's gene 2 that's turned on at first, and then eventually turns off, and then gene 1 turns on. But yeah, the important thing is that they're not really on at the same time. They kind of like both turn each other off. So that's kind of the point of the toggle switch model of gene regulation. Yeah, I kind of picked these parameters to get that nice result, but I encourage you guys to like change these parameters around, like experiment with them, see what happens when you change them up. But yeah, so that's basically all I have for this video. Again, any questions, just let me know in the comments. I'm gonna put the code. I'm gonna put the code on my GitHub if you guys want to download it and try it for yourself. But yeah, so thanks for watching, guys, and see you next time.